Hello everyone. Today I give a lecture at Advancing Nuclear Medicine Imaging 1. In this course, as a part 1, I will talk about radionuclide tracing technique. First, we should know the definition. Radionuclide tracing technique means the radionuclide or its labeled compounds as tracers detected with the radioactivity detection instruments could be used in the study of the objective existence and the change of tracers in biological systems or external environments. Radionuclide tracing technology is a basic and the most important technology in nuclear medicine, and it is also the methodological basis for the application of radionuclides in the medicine and biology and other areas. Radionuclide tracing technique needs two factors: tracers and radiodetective instrument. First, tracers. We also should know the definition. What is tracer? Tracer, a radioactive tracer or radioactive label, is a chemical compound in which one or more atoms have been replaced by a radioisotope. What's the application? There are two main ways in which radioactive tracers are used. When a radioisotope-labeled chemical compounds undergoes chemical reactions, one or more of the product will contain the radioactive label. Analysis of what happens to the radioactive isotope may provide detailed information on the mechanisms of the chemical reactions. And another application is a radioactive compound is introduced into a living organism, such as human body, animal, and the radioisotope provides a means to construct an image showing the way in which that compound and its reaction products are distributed around organisms. Actually, radioisotopes of the hydrogen, carbon, phosphorus, sulfur, and iodine have been used extensively to trace the path of the biochemical reactions because these elements belong to the most common elements in our body. A radioactive tracer can be used to track the distribution of a substance within a natural system, such as a cell, a tissue, as a flow tracer to track the fluid flow, such as blood volume. Radioactive tracers are also used to determine the location and function of a definite molecule, as a radiocarbon dating used for natural organic carbon-14 isotope as an isotopic label. Second important thing is radioactivity detective instruments. Radioactive tracers could be detected by a radioactivity detective instrument. For example, gamma counter to count the gamma ray, liquid scintillated counter to measure beta ray, gamma camera, SPECT, PET, in vivo radio image to form the image. Radio tracers are the basis of a variety of imaging systems, such as PET scan, SPECT scan, gamma camera. So it's very important. Next, I will talk the principle of radionuclide tracing technique. There are two principles. First, identity of labeled and non-labeled compounds. An atom in a chemical compound is replaced by another atom. They are the same chemical elements. Sometimes they are similar. For example, when we labeled a peptide or protein with iodine 125, replaced a normal iodine 127 by iodine 125, normal iodine atom and iodine 125 show the same features. Another example, we use a tritium, usually we call H3, replace hydrogen, we call H1. The tritium and hydrogen show the same features. However, the substituting atom is an radioactive isotope. This process is often called radioactive labeling. We labeled something with radioisotope, so this process is often called radioactive labeling. Second rule is the measurability of radionuclides. Radionuclide and its compounds can give out a variety of gamma, beta, or alpha ray, and can be measured by radioactivity detector or recorded by the photosensitive material. They made a precise qualitative, quantitative, and location information about labeled compounds, so we can carry out definite measurement and research. The measurement instruments include in vitro gamma counter, liquid scintillator counter. In vivo, we have the gamma camera, SPECT, and PET. Radionuclide tracing technique is so important and powerful. The power of radionuclide tracing technique is due to the fact that radioactive decay is much more energetic than chemical reactions. Therefore, radioactive isotope can be present in low concentration, and its presence detected by the sensitive radiation detector. Now I talk the characteristic of radionuclide tracing technique. First is high sensitivity. They can detect a very low amount of substance, lower than nanogram. The second, the detective method is simple, accurate, and does not need to be separated. You detect it with radioactivity detective instrument. You don't need to isolate A from B. You can detect radioactivity for a compound, complex compounds. The third, reflect physiological condition. The amount of chemical required are very small, does not disturb or destroy the balance of the physiological process in the body. They will reflect actual situation of the body. The last, combination of qualitative, quantitative, and position studies. So with this technique, you can know the radioisotope, where it is, yes or no, or how much. There is the most important thing you need to remember. The biological behavior of radionuclide tracers in vivo mainly depends on the labeled substance, not radioisotope. It's radioisotope labeled substance, where the radionuclide only emit radiation during the metastatic transformation of tracers, just shows the role of the tracer. The same radionuclide labeling on different compounds shows completely different metabolic process and biological behavior in vivo, while different radionuclide labeling on the same compounds will not change their biological behavior. Common types of radionuclide tracing technique in vivo includes the following. First, tracing the material, metabolism, and transformation. Second, cytokinetic analysis. Example, cell cycle measurement. From G phase to M phase to S phase, so different phases. The third is in vitro radioassay, such as radioimmunoassay. In vitro tracing technique, also known as in vitro radioisotope tracing technique, they take sample system such as tissue, cell, body fluids. They separate it from the whole body as a research object. It is mostly used to study the transformation of certain specific substance, such as protein, nucleic acid, cytokinetics analysis, activation analysis, and in vitro determination of very low amount of substance. In vivo, they include the following: absorption, distribution, excretion of substance with radionuclide tracers, radionuclide dilution method, autoradiography, determination of organ function with radionuclide tracer, radionuclide tracing image. In vivo tracing technology, also known as in vivo tracing technology, they take the complete organisms as a research subject. For example, animal, human, other organisms. They used to study the qualitative, quantitative, and localized dynamic change of the absorption, distribution, metabolism, and excretion of labeled chemical molecules within the biological system. In vivo tracing techniques are used on dynamic analysis. Here, we may pay more attention to in vivo study. Okay, this part we talked here. Thank you.